welcome back. My marker is refilled. Today, we're talking about respiration. We'll talk about some basics, and then we'll get specific with aerobic respiration. And in the next video, we'll talk about anaerobic respiration. Okay, so respiration, we have an electron acceptor. So that means we're going to be transferring electrons, right? So we're going to be talking about electron carriers. And you probably have heard about, if you've watched any of these videos, you've heard about the electron shuttle NAD+, or when it's reduced, NADH. So NADH will donate electrons to compounds to reduce them. And it can also donate electrons to the ETC. So these are the two things that it'll carry electrons to. And there's another compound called FMN, and this will also carry electrons and electron transport chain. So it's an intermediate electron carrier in ETCs. Okay, that's cool. And we have another one. Next one we're going to talk about is quinone. And we'll talk about this more when we talk about the electron transport chain. But this one is mobile, so it can move, and it's also hydrophobic. So you'll usually see it in membranes. In fact, that's all you really see it in. So these three different categories, we have NAD, NADH, FMN, and quinone. All these three categories can carry 2 electrons and 2 protons. So it might not seem like NADH is carrying 2 protons. You can see how it might carry 2 electrons, because if you have the plus, if you were to carry 1 electron, it would make it neutral, and then another one is offset by the proton. But if you were to see the structure, the oxidized form has 1 less hydrogen here, and then there's another hydrogen on the side. So those are where your 2 protons go. But there are some that don't carry 2 electrons and 2 protons. For example, cytochrome. So cytochromes are proteins that contain a heme. So what is a heme? A heme, it's a porphyrin ring, which you don't really need to know what the structure of this looks like. But you do need to know that in the center of the ring, it contains iron. And another thing containing iron is iron-sulfur clusters. So an iron-sulfur cluster is usually depicted as like a cube of iron and sulfur, and it's kind of alternating. I'm not going to draw it like that because you don't really need to know the exact structure, but you might want to know that it is connected to a protein. So it's kind of similar to a heme in that way. It's connected to some sort of protein in the electron transport chain, right? So that's what this is. Protein in ETC. It's covalently bound with cysteine residues. So there's going to be the amino acid cysteine, and I'm just going to write it as cis, so if you want to mark that as something else, you can, but I'm just going to write cis. So it's bound to this protein in the electron transport chain, connected to the cysteine amino acids, okay? So that's what's happening here. So the way that these three had some stuff in common, cytochromes and iron-sulfur clusters have stuff in common. And actually, I wouldn't even say that it's the cytochrome and the iron-sulfur clusters that have stuff in common. It's really the heme that is our electron carrier here. The cytochrome is more like the protein that it's bound to here. So I'm actually going to highlight heme, and I'll just underline cytochrome so it can be consistent with the way that we're looking at things here. Okay, so for both of these, they're going to have iron. That's iron 3. And when it gets reduced, plus electron, turns into iron 2. So both of these irons, the one in the heme and in our iron-sulfur cluster, will get reduced to iron 2. Another thing is, remember how the iron-sulfur cluster is covalently bound to this protein? Well, same way, the heme is covalently bound to the cytochrome. So this is the thing we have in common. Covalent bonds between protein and electron carrier. So the heme and the cytochrome, and the iron-sulfur cluster, and the protein in the electron transport chain. And whereas these all carry 2 electrons and 2 hydrogens, both the heme and the iron-sulfur cluster only carry electrons, right? If we're just reducing iron, then that's not going to carry a proton with it. Okay, let's write out some qualities of electron transport chains. So ETCs. So we know kind of the different carriers that are involved in electron transport chains and, like reducing compounds as well. But as a general rule, it's really important to remember when we learned about that standard reduction potential, that's a pretty general rule. That's a pretty general rule in biology, right? So of course, electron transport chains, where reduction and oxidation is happening all over the place, electrons will only be transferred, or will always be transferred, from a low standard reduction potential, or E naught, to a high E naught. So that's favorable, right? That would release energy. And then electron transport chains, they span a membrane. Because if you think about your cytoplasmic membrane, you're storing energy as your proton gradient, so your proton motive force, and you can't have a proton gradient if there's no barrier. If it's just out in the open, there's nothing going to keep the protons in. Another cool thing is that some bacteria can do multiple types of respiration. And we'll learn about this when we learn about anaerobic respiration. But anaerobic is generally less efficient than aerobic respiration. So bacteria that can do both will want to be able to shift back and forth when environment changes. So in the presence of oxygen, you're doing aerobic respiration, and without it, you can do anaerobic respiration. So the electron transport chain units, or subunits, so like your certain protein complexes, or the specific electron donors you use, can be swapped out to adapt. So that's really cool. It can make sure that they're always doing the best thing considering their environment. Okay, I know this page isn't that full, but we're actually going to switch to the next page because we're going to do a sideways thing. So with our electron transport chain, we're going to be moving across a membrane, right? And we're going to be transporting the electrons that we get from our early reactions. So if we did glycolysis, which I assume we have some NADH from that, and also from the citric acid cycle, because with aerobic respiration, you do that. And aerobic respiration is done by chemoorganotrophs, right? We get our electrons from an organic molecule, and our chemical energy from organic molecules as well. So we're going to start with this membrane. You can imagine this side as the periplasm of a gram-negative bacterium, and this is the cytosol. So we have NADH from catabolism, right? We did glycolysis or the citric acid cycle to generate NADH, and we are going to use its electrons. So we are going to use its electrons, and that will strip off its protons. So if we're taking off the protons, the hydrogen ions, maybe we'll go through an enzyme called NADH dehydrogenase, perhaps. So that's what this is. This is NADH dehydrogenase. So we're going to split off the electrons and protons from NADH. So that'll make NAD plus again. And we're going to retrieve our 2 protons. And that is going to reduce FMN, that electron carrier we were talking about before. So it's going to take those electrons, and actually, let's draw the electrons as red. So these are our electrons, so we can keep track of our different reactions. So this will yield FMNH2 because it's been reduced, right? I'm just going to draw this red line as like an arrow of the path of the electron. So it'll get passed on to the iron sulfur clusters that are in NADH dehydrogenase. And then remember, this is all through a series of redox reactions. So as these electrons are passed along NADH dehydrogenase, we actually can push 4 protons against the gradient. So this is the acidic side, right? We're in the periplasm, so the concentration of protons is high here. It's acidic. And then here it'll be lower. So this requires energy, and I'm going to highlight these protons just so that we can keep track of them. Okay, so how did we do this? How did we push these 4 protons up the gradient? So protons are pushed up the gradient because we release energy from these redox reactions. So using energy released from favorable redox reactions. So favorable meaning there is a positive delta E naught prime. So NAD is going to have a lower reduction potential than FMN, which is going to have a lower reduction potential than our iron in our iron sulfur clusters. And then as it gets passed along in these different molecules, those will all have increasing reduction potential. So we're going to be releasing energy every time we do this. And we can use that energy to push up the protons. And the way that we do that is the transmembrane proteins involved in NADH dehydrogenase, the like physical protein, they have side chains that can get protonated. And then they can be deprotonated. So that means that a proton can travel along one of the proteins until it's pushed out into the periplasm. So protons are pushed up the gradient using energy released from favorable redox reactions, meaning a positive delta E naught, and the protons are pushed along transmembrane protein side chains that get protonated and deprotonated. Okay, so our electrons are now here, and what we're going to do is we're going to reduce a molecule that's actually closer to the cytosol side of the membrane, called quinone. And so if we're reducing it, that means we are adding electrons, so we're also going to need protons. So there's 2 protons involved here. We'll highlight those as well. And we're going to make a reduced quinone, our hydroquinone here, with our 2 protons. And now, and that can go to our cytochrome BO complex. So we're going to have our proton pump here. So this is our cytochrome BO complex. So this will be site B, this will be site O. I'm actually going to draw this a little bit fatter. And it'll bind to cytochrome B and get oxidized again. So we'll go back to normal quinone, which doesn't have our protons. And in the process, the protons will get pushed out, and that'll actually contribute to the proton gradient. So what we did here is we passed the electrons on, and since cytochromes, which remember they contain hemes, right? So there's heme B here, and so our electrons are going to pass through the heme. But the hemes don't carry protons, remember, they just carry electrons. So since we can't carry the proton, that's fine, because this way we can export it into the periplasm, and that'll contribute to our gradient. And then this will get passed on to heme O3. So site B is for heme B, and site O is for O3. And there's also a copper atom here, and our electrons will be accepted by the final electron acceptor. So we'll start, and if we're doing aerobic respiration, our final electron acceptor is oxygen, and we'll use 2 protons again to make H2O. And this is kind of contributing to our proton gradient in the way that there's going to be less protons here, but there's not going to be more protons on the outside because it gets converted into H2O. But I think that's interesting. So we have quinone, it gets reduced to hydroquinone, and also there's a bunch of quinone molecules in here. It's called like the quinone pool, so they'll like be cycling through here all the time, because now this one can go back and retrieve more electrons and continue to do this Q loop. But we're just drawing the cycle of this one molecule. So yes, our electrons get accepted by the final electron acceptor. The electron transport chain is over, but we do still have one important component left. Now that we've built up this great proton gradient, it's very acidic out here. There's so many protons. You know what we got to do? We got to make some ATP. That's what all this work was for. So there's 2 components of our ATP synthase enzyme, and we'll go over the subunits of these 2 components a little bit. But maybe you learned in Gen Bio about those 2 units, the F0 and F1 subunits. So one is the part that is embedded in the membrane, and one is the part that actually synthesizes ATP. I'm just going to draw like kind of basic diagram here. Okay, so this brown part, this is F0, this is embedded in the membrane. You can remember it because it is not the part making ATP if you want. And this part is F1. This is the part that will actually be synthesizing ATP. So when we let in a proton, and it goes through here, and what happens is there's part of this F naught complex that is the stator, so that is stuck and it is not moving. The part is the rotor. So when a proton travels through, it'll push on the stator and move the rotor, and that'll cause it to rotate. And then there are components in F1 that will change conformation as the rotor is spinning, which will bind with ADP and make ATP. So this process does what kind of phosphorylation? It's not substrate level, it's oxidative. So we'll be making ATP through oxidative phosphorylation. So let's go over what I was just talking about. So the F naught part is composed of the stator, and two parts are involved with that. That's parts A and B of F naught, and the rotor, which is part C. So when protons flow through, they flow through A, which, remember, the stator is stuck to the membrane. It's not going to be moving, but it'll push on C, and that'll cause it to rotate. And then we have F1, and there are two parts that are bound to C. So epsilon and gamma are bound to C, and since C is spinning, they also spin. And this causes a conformational change in another two components. So epsilon and gamma are part of F1, and so is alpha and beta. And I think it's nice because you can remember F naught, all of these are like normal alphabet letters, and this is the Greek alphabet in F1. So alpha and beta will have this conformational change, and that'll drive ATP synthesis. So driving ATP synthesis. So that's what we see here. So you don't have to memorize like on this diagram what each part looks like, but you do need to know that here I'll highlight it with the right colors. So A and B are stator. They are not moving. The rotor is C. That is moving. The stator is pushing on the rotor. And if you need a review of these terms, watch the flagella video, the bacterial appendages video. So when protons flow through A, it pushes C, and then that causes it to rotate. And since C is rotating, so C is actually this bit, so this is A and B. These are like stuck to the membrane, they're not going anywhere. C is also part of F naught, but it is rotating, and it's taking this thing with it. So as C is moving, it's pushing the F1 complex too, and that's including these two pieces, epsilon and gamma. So they're bound to C. They also spin, and that causes a conformational change in alpha and beta, and that drives ATP synthesis. So we passed along these electrons, we moved a ton of protons, and actually we moved even more protons, because this is a proton pump, right? So we took the protons that was attached to hydroquinone, and we pushed those against the gradient, but we also, using the electron transfer in these redox reactions, were able to push out two more protons. Forgot to mention that. So we're really contributing to our proton motive force here, which is why we're able to use that to synthesize ATP. So, all right, let's do a review. So these are our electron carriers. We have NAD and NADH, and then I guess I never wrote it, but if you wanted to write the reduced form of FMN, it would be FMNH2, and then quinone, like you have Q, you can say Q sus QH2. So these three molecules all carry 2 electrons and 2 protons. That's important. So NAD and NADH can donate electrons to compounds or the electron transport chain. FMN is mostly an electron transport chain carrier. Quinone, it's mobile, it's hydrophobic, and that's also something that we see in our electron transport chain, like in the membrane. Cytochrome is also part of the electron transport chain. It's a protein that contains a heme in it, and a heme is a porphyrin ring that contains iron. You don't really need to know what a porphyrin ring looks like. But also something that contains iron is our iron sulfur clusters, which are similarly to a heme, connected to a protein in the electron transport chain. So this exterior protein is very similar to cytochrome in that way, in that inside it has this electron carrier molecule with iron, and in both of these, iron 3 gets reduced to iron 2, and they only carry electrons. They don't also carry protons like the other ones we were talking about. And our electron carrier with iron here are connected with covalent bonds to the protein in the membrane. Okay, and then some general principles of electron transport chains. Electrons will always be transferred from low reduction potential to high reduction potential. So it has a greater potential to be reduced. So that's favorable. In an electron transport chain, this is an example of something that's favorable, so you get electrons from it. You can push things up, if necessary, to a lower reduction potential, but that requires energy, and in a reaction here where we're trying to get energy, that's not going to happen. Electron trains will always span a membrane, and they store energy as proton motive force, because if there weren't a membrane, you couldn't store any protons in a higher gradient in one place or another, because otherwise they would just float in the lower spot. Some bacteria can do multiple types of respiration. And we'll learn more about that when we learn about anaerobic respiration. We'll use nitrogen instead of oxygen in certain situations. So electron transport subunits can be swapped out to adapt, like complexes or electron donors. Okay, and then we have this beautiful figure, our diagram of aerobic respiration in our electron transport chain. We have our enzyme NADH dehydrogenase here, which pushes 4 protons into the periplasm, and it employs that FMN that's in the electron transport chain, the iron-sulfur clusters, and then a series of redox reactions that release energy in a way that can push protons up the gradient by protonating and deprotonating the side chains of certain transmembrane proteins. And then that'll push it out, and you'll get 4 in NADH dehydrogenase through all of those reactions. And then the electrons will be transferred to quinone. It'll take another 2 protons that get exported from the cell, and then cytochrome B with heme B will push another 2 protons. Depending on the model used, sometimes you'll see another 2 protons being exported here. It depends. Don't worry about it. So the electrons will travel through here. They'll get accepted by our final electron acceptor oxygen, with some protons, and then turn into H2O. And all of these protons, so here we see 8, some, like I said, some will show you 10, but either way we have lots of protons in here in the periplasm, and we can use that concentration gradient to let out one and make some ATP. And fun fact, it's like 3 to 4 protons per ATP made. So that's how many protons you have to use to let out through the gradient to make one molecule of ATP. So it's not just like 1 proton makes 1 ATP. It's about 3 to 4. All right, well, hopefully that made sense. That was our aerobic respiration, and next time I'm going to catch you for anaerobic respiration, so stay tuned for that. Okay, bye!